Hello there and welcome to this Khan Academy tutorial on AQA A-Level Biology. And in this tutorial we will be looking at DNA and RNA. So the aims of this tutorial are to give clear and concise information, and all information given in this tutorial are linked to the AQA specification. The words marked in bold are the key mark scheme words, and all information in this tutorial are as accurate as possible. And if there are any errors that are found, please notify in the comment section below so it can be corrected as soon as possible. These are the AQA specification points that we will be covering in this tutorial, so let's get started. So let's start with answering what is DNA, and DNA is a molecule that stands for deoxyribonucleic acid. It is a polymer consisting of nucleotides, and it stores genetic information and that is located in the nucleus. So the nucleotide consists of a hydrophilic phosphate, a deoxyribose sugar, and a nitrogenous base. Because it has a hydrophilic phosphate, DNA is negatively charged. The phosphate forms the backbone of the DNA and hence giving it a negative charge. DNA is a polar molecule and is soluble in water. The nitrogenous base of DNA consists of adenine, thymine, guanine, and cytosine. Adenine's complementary base is thymine, while guanine's complementary base is cytosine. Let's now look at the structure of DNA. And the DNA's nucleotides are bonded by condensation reaction. So the nucleotides are joined together by condensation reaction to make this DNA, and the way they do this is the phosphate on the 5' carbon reacts with the hydroxyl of the 3' carbon, forming a phosphodiester bond. And the formation of this phosphodiester bond is catalyzed by the enzyme DNA polymerase. DNA is considered to have a right-handed double helical structure. You do not need to know this information for the specification, but this is additional knowledge that will be useful for the essay. The two strands that make up the DNA are considered to run antiparallel. So the phosphodiester bond in this DNA strand is located here, and the right-handed helical structure that I was talking about is shown in this diagram. So it's basically how the right hand would turn, forming a right-handed helical shape. So let's have a look at these antiparallel strands of DNA. Note that one strand runs from the 5' to the 3' and is called the lagging strand. And the strand that runs from the 3' to the 5' is called the leading strand. The reason why they call 5' and 3' relates to the carbon number. So the 5' refers to the 5th carbon in that pentose sugar, whereas the 3' refers to the 3rd carbon of that pentose sugar. DNA replication, the lagging strand and leading strand are important in DNA replication, and this will be covered in the next tutorial. So adenine's complementary base is thymine, and there are 2 hydrogen bonds that link them together, whereas guanine and cytosine have 3 bonds that link them together. Now, this replication of DNA we will not be looking at a great detail, as this will be covered in another tutorial. And the main summary is that we say that DNA replicates semi-conservatively. And what that means is that the resulting DNA consists of an original strand and a new strand. The original strand acts as a template so that the new strand can form. And because of this, genetic continuity is ensured. And this also allows for accurate replication because it replicates semi-conservatively, and the complementary base pairing ensures that there are as little errors as possible when DNA is replicating, reducing the risks of diseases such as cancers. Now, there's also the weak hydrogen bonds, which are also enabled to carry out an accurate replication. And before the scientists realized that DNA replicated semi-conservatively, many believed that it replicated conservatively. And what that means is that the resulting DNA strand had completely 2 new strands rather than an original and a new strand. So they devised this experiment where they had a heavy DNA consisting of the base, where the bases had the heavy nitrogen, so N15, and added this to a light DNA where the bases had the 14 nitrogen. So nitrogen 15 and nitrogen 14 are isotopes, so they have the same atomic structure, but they differ in the weight. So when they added, they found the result that the resulting DNA consisted of N14 and N15 rather than two bands of N14, N14, and N15 and N15. And this was symbolic of semi-conservative replication rather than conservative replication, which would have meant there would have been two bands. And to confirm this, they added this resulting solution to N14 and N14 again to the light DNA solution, and the results were as expected. Is two bands of N14 and two bands consisting of N14, N14, and then N14 and N15. The N14 of the right, N14 from the light DNA would react with the N14 from the heterogeneous DNA, forming N14 and N14, and the N15 from the heterogeneous DNA would react with the N14 of the light DNA to form N14 and N15. So the height of these bands indicate the weight of the DNA, and the thickness relates to the proportion. So the thickness would decrease for the N14 and N15 as the reaction progresses, whereas the N14 and N14 would increase in proportion. So the history of DNA is not needed to be known at a great detail, but I have summarized the main points. The DNA was observed around the 1800s, and many scientists believed that proteins carried the genetic information because they were complex and varied. They had many different primary structures, so they were much bigger than DNA molecules, so they had to carry the genetic information. However, this is untrue, as they figured out later, and the double helical structure of the DNA was determined by two male scientists, James Watson and Francis Crick, using the evidence of X-ray crystallography, which was given by a female scientist called Rosalind Franklin. So the X-ray crystallography with the characterized X shape indicates that the DNA must have a helical structure. So how the DNA structure helps its function is a classic exam style question. And to understand that, we have to look at the sugar phosphate backbone, which provides its strength. The phosphodiester bonds enabling to form the sugar phosphate backbone are very strong, which means that they can protect the bases and also provide strength to the DNA. DNA is a compact molecule because it's coiled, and because it's a very long molecule, a single strand can store a lot of information. The complementary base pairing of DNA allows for an accurate replication, like I said before, and this also enables DNA to carry out its function very well. And the weak hydrogen bonds, like I said before, means that the bonds can be broken easily for DNA replication. So now let's have a look at the structure of RNA, which has a similar structure to DNA. However, it's a single short polynucleotide strand rather than a long double polynucleotide strand. The RNA basis contains of adenine, which has a complementary base uracil, and guanine, which has a complementary base cytosine. Again, the number of hydrogen bonds stay the same, the two bonds between adenine and uracil, but RNA does not have thymine, but uracil and thymine have a similar structure. Again, the RNA nucleotide has a hydrophilic phosphate, so RNA nucleotides are polar, usually having a negative charge. And the enzyme responsible for the formation of RNA is RNA polymerase. The RNA has many functions in our body, and some of them are protein synthesis, gene silencing, DNA replication, and making the functional units of ribosomes, which consists of two subunits. And all these will be covered in the next few tutorials. So this table summarizes the differences between DNA and RNA, which again is a common exam style question. And with the end of the tutorial coming up, I would like you to pause the video and have a go at these exam style questions. And these are the answers for those questions. I'm sure that you would have realized that all the information in this tutorial are almost exactly the same as these answers, because all the information that was generated were from many mark schemes. So the information given in these tutorials are as accurate as possible. So if you do find any information, do notify in the comment section. So thank you for watching the tutorial, and if you for more free resources, go to carbonacademy.wordpress.com, and thank you very much.